Okay.
Esteemed keynote speakers, colleagues, participants, ladies and gentlemen, good morning and good afternoon to all. I am Fatma Zehra Çağıl, doctoral researcher at Dr. Metin Rafiki Lab. I will be hosting for the second day. I would like to announce some housekeeping messages regarding our symposium. To ensure a smooth running of the workshop, please make sure you have access to a good Wi-Fi or cable internet connection. We kindly request that you change your Zoom profile name to your full name to ease name tracking for attendance purpose. The participant certificate will be only issued upon attaining all 3 days attendance. The sessions will be live streamed on YouTube. For those who cannot get into the Zoom due to any technical issues, please join there and inform us. Please join our Discord server for announcements and other communication. If you have any technical issues, kindly seek help through the chat box or email to us. If you'd like to ask questions to our speakers, please type them in the chat. Now, I would like to welcome our chairman for this morning keynote talk session, Dr. Metin Rafiki. I hand over the platform to Dr. Metin, who will continue from here. Over to you, dear Dr. Metin.
Thank you, Zahra. We're going to welcome everyone to this session of the symposium. The session first speaker is Dr. Pawel Suwinski. I'm going to introduce him briefly. Dr. Suwinski is a medical professional. He studied at Medical University of Warsaw, and he did, but afterwards he moved more into healthcare management, did a master's in Swansea University. Currently, he's working in the Malaysian Genomics Resource Center, Berhad, as a clinical geneticist and medical bioinformatician in Kuala Lumpur, Malaysia. He's going to talk about personal pharmacogenetics and how it influences treatment plans. Professor Dr. Suwinski, the floor is yours.
Thank you very much, and good morning to everyone, or good afternoon, depending where you're connecting from. And just one correction, not working anymore with Malaysian Genomic Resource Center. I'm working for actually my own company, MyGenix, that does bioinformatics, or the secondary and tertiary analysis of clinical data. I mostly deal with humans. I'm a medical doctor. I was practicing, so I use genomic information to improve clinical outcomes. And my past, I think, 11 years was basically to try to find a better way to use the information and how we can actually utilize tools that we have, which are the IT tools. And I like to compare my work, or bioinformatician work, to ice hockey player. I'm not sure whether you ever watch how they play. It's a very fast game, and you don't even see that they have a skate on the boots. When they look at the pack, where it goes and whether they score the goal. So the same with bioinformatics. The IT actually skates. They are the tools to get us where we want. So let me start with my presentation, because I understand the time is of essence and we have a very limited time. I have to share my screen and I have to find my presentation at this point. Yes, it's over here. So let's go on. During the course of the, I'm trying to make it, yes. During the course of the presentation, I will be showing you some files. So I hope I can switch between the sharing of the screen to a few other files that I would like to show. although I'm a clinician and although I will be talking about clinical application of pharmacogenetics, this is bioinformatics workshop, and I will be also including certain areas of bioinformatics and how to go about to analyze genetic profile towards your response to drugs. So, first of all, if you are having, if you are actually seeing that whatever my agenda is not in line with what you would like to hear, just voice it out, raise the hand or give the chart. I will adjust. The agenda is quite, as I said, quite ambitious, and I'm not sure whether you can see the screen. Do you screen, or I have to share it with you?
Right. Oh, we can see the screen, Dr. Suwinski.
Oh yeah, you could actually see that one. This is my agenda. These are very, very ambitious, and we will be talking about many things, and I hope you will learn a bit and something practical, because that's what I do, and apply in your work, or improve your improve your studies. Now we're going back to my presentation, and let's go to the next slide. For different people, or various people, the pharmacogenetics or precision medicine means different things. But originated, or the original meaning was pharmacogenetics. The reason why pharmacogenetics is because this is the only area of genomic information that is precise. The rest deals with uncertainty and prediction. So this was the first area fully accepted by conventional medicine and is practiced for the past 10 years. Over the time, it evolved because we had better tools, and we understand better the genomic information, and we started expanding the scope, and it started becoming closer to personalized medicine. It's not personalized medicine, but it's big chunk of personalized medicine. I'm subscribing to the bigger scope, and that's why I will be talking many areas of the precision medicine, but of course focusing mostly on pharmacogenetics. So at its core, at its actually basic or nutshell, precision medicine refers to an individualized treatment that improves clinical outcomes. What it means, improve clinical outcomes? It means that whatever was planned and will be better. It means that it changes the treatment, and the patient will either recover faster or be cured. So this is where we talk about clinical outcomes. What are the components? Is predictable. We want always treatment to be predictable. Right now is a trial and error. It is 50-50 sometimes. So when I was a young doctor and a patient came to me, I wanted to say actually on the door, I only know one disease. But anyway, when they came and I gave the drugs, they said, if it doesn't work in 3 days, please come back. So this is where we don't want to have it. We want to have predictable. I mean, we want what will happen when we give. Individualized genomic information is the only one that can do it. Precise. It means it only acts on areas that we want. And of course, high clinical utility, it means that the improvements outpace and are economical and outpace the conventional treatments. And as I mentioned earlier, it uses genomic information. So down the last sentence actually is summarizing it is not only pharmacogenetics, but it's also disease risk prediction that is using genomic information. That slide tells you, it's busy, but it tells you exactly what I mentioned. I want to have a precise medicine. I want to have a predictable. So this is predictable. This is precise. This is the information I'm getting. This is the black box that we're using. Some deploy artificial intelligence, some they own algorithms, and then of course right on target, predictable. Now we are moving from intuitive to evidence-based and actually precision medicine. We're not looking at symptoms anymore. We start finding the causes at the molecular level. Genetics always will be underlying cause of every disease or problems, or actually who we are, every trait, with the color of our hair, eyes, or let's say whether we can tan faster or have a curly hair. How do we use it? In the broader scope from pharmacogenetics. So this is the early use. Pharmacogenetic test, we're looking at drug metabolism, drug response, and adverse drug reactions. Then we evolve into predispositions when we're looking at cancer targeted treatments, and we're looking multifactorial diseases. When I say multifactorial diseases, they are also genetic based, like, for example, diabetes type 2, monogenics. Mental traits, they are highly heritable. Sport medicine, we're also looking at that. This is precise medicine. We can actually train athletes, elite athletes. And then we're looking at something that is called diagnostic test. This is 100% predictable. Once you have that particular variant in the gene, you have the disease. Utility is actually very low because you can't do much. And utility, the highest one is in pharmacogenetics, clinical utility. Medium range utility will be for predisposition and prognostic tests. So that is what will happen in the future. I was actually in a roundtable a few days ago, and they were asking what will happen in the next 5-10 years. Genomics will be at the center because we will have to have genomic information and measure it up together with all other information to derive the full picture, and it can be deployed in outreach or in the schools or screening, prevention, and of course, now we can call it genomic medicine. You have to understand that genomic information is the only tool that helps us with prediction. There is no other tool that we can deploy. It's only gut feeling, or let's say family history. Let's go to the next slide. Now we start bioinformatics, just a small bit. This is how we get the information. And we get the information to do pharmacogenetics, all the other beautiful tests. We use two major technologies. They're more, but they are two major. Of course, sequencing, and you see on the right side, which actually have a sequence of all your letters, whether it's a gene or let's say the whole genome, or we can look at genotyping, which actually looks at specific locations that already are proven in the literature and in, I would say, databases as highly pathogenic, which might be in a ClinVar or ACMG. So we don't have to do sequencing. We look for particular variant. It can happen, for example, in sickle cell, where there is only one mutation, or on lip and down disease, when there is one gene and few mutations. So that's where we have to understand where to deploy sequencing and where to deploy genotyping. For example, in thalassemia beta, we can genotyping we have to deploy sequencing because we do have de novo mutations. And the one disease, genetic disease, that always will have de novo mutations, not acquired from parents, it will be the Soto syndrome. So knowing the patient, we will choose the right technology, and then we acquire the information. And how do we do that? What is the next? The next is actually a very rough story. We are not precise. Maybe Oxford Nanopore can do one strand or genome, but they still struggling with quality of that. So we're still using the millions of strands after blasting few million of cells and then creating those confetti shreds that later the machine optically will put into the sequence of those letters. This is very simple on the left side for the sequencing. And then it comes the magician, which is a bioinformatician. The magician will take the letter, which actually is a total gibberish. You don't really know what it is. And it will know after many, many hours of working and using hundreds of tools, what are the variants or what happened to that letters and the meaning of the letters, and then advise the clinician what to do. And this is really the role of bioinformatician, and I like it very much because it's exploratory. Sometimes every patient is different, and it is not a routine job, and you have to deploy many, many tools. In fact, myself, I do create my own tools, and I will show you later how actually it works. Let's go to the next slide. Okay, so this is the magician guy. He gets the information from sequencing, from transcriptomes, and from, let's say, methylation or epigenetics, and we'll start looking at any possible changes. And this is on the right side, that he, according to his judgment. Now, that you have to understand that the first two columns are pretty much standardized, but the third and fourth columns is Wild West. It's up to the experience of the bioinformatician or clinician, or the proper communication with the clinicians, and understanding the patient, that you'll know what to do and how to interpret what it was found. So this is the three levels of how the magician or bioinformatician is finding the variant and putting a meaning to it. As I said, the primary processing is already well established, are using GATK plus well, other tools. There is around 30 plus different programs and steps and many programs to actually get your FASTQ file, which is the very raw file that comes from the sequencer, and then end up as VCF file. VCF file is a standard file established by 1000 Genome, and it is being used for next steps. This is a standard file, and everyone should actually get the file before going forward. The secondary analysis is molecular annotation. When I say molecular, it means we actually want to know what is the molecular effect. It's nothing to do with clinical effect. We're talking about molecular effect, where the variant is actually positioned, intron, whether it's pathogenic, what is the impact, whether it's medium, low. And then we're using in silico algorithms to understand better the pathogenicity of particular variant, especially if they are new ones that we cannot find in primary and secondary databases like ClinVar or GenBank. So then we're moving to the third one, which is knowledge-based prioritization. And that's what I said, this is the Wild West. It's up to the person or bioinformatician to deploy tools. My experience, and I will show you later, certain off-the-shelf tools are not useful for me. They generate a lot of metadata, which means is unnecessary. I'm actually looking at certain particular information. I don't want all this, I would say, enormous amount of information additional that are not useful to me. And I have to deploy another tools to clean and sieve it or filter through it. So mostly I will use my own scripts and applications to do the knowledge-based prioritization. You have, when we say knowledge-based prioritization, it's not just the application, because the application are used to connect to appropriate databases and pull the information that I need. If it's pharmacogenetics, I just need, for example, PharmGKB database and pull out of it all the information that is relevant to particular variant or particular, let's say, DCF file. Let's go. Next one. So the first and second standard, stages are, as I said, pretty standard. I'm showing you the G80K golden standard. This is older files. Probably they have now 4.2 version, and they integrated together with another application, which is Pickup. This is mostly Java-based, but they still use other BCF tools, SAM tools, additional tools, and they annotate with dbSNP from NSBI. So you can annotate during the process, but this is the bottleneck. You have to understand that this is actually the bottleneck in the whole process. It takes up to 2 days. It depends what you want to do. Up to 2 days. So I was talking to, and yeah, you can use a cloud computing, but on a long term, it won't actually save you money. It will save you money in a short time, but in a long time, your storage actually would be a very problematic issue. And clinical data is not readily acceptable to be in a storage. All hospitals would like to have it locally, and so you need application as a service, and normally hospitals will have their own platform, which is called high computing performance. And you have Intel and, for example, Lenovo right now coming with something that, okay, when I said it can run in 2 hours, they can actually shorten it to 15 minutes. Of course, you pay for it, but your turnaround is now very fast, and you actually can do it as a point of care. Let's go next. This is the Wild West. These are old slides. I didn't put all of it, but the next slide will give you some idea what other filters or applications are in the tertiary databases or for prioritization of variants. What we see here is actually primary databases. Then they have their own tools. We have we have the algorithms in silico and databases, but we can get information. I will be talking about them in a while. So these are few years ago I created to tell people what actually is happening, what I do. As I said, the first and second stage is pretty simple. I get GTK, I can link to ANOVA, I can link to VEP and actually get molecular annotation. What comes next is, I have molecular annotation, but I don't have the clinical annotation, and that's where I start doing it. I have to look into networks. I have to look at the genetic ontology. I have to look at the connections, which is reactome, biases. I will show you if you want at the end, and I have in slides examples. And for genetic diseases, which you actually have to have a clinical grade, you need to use application like TGX from LifeMap. They changed the name. Right now they are Genix. I will show you Genix. I will show you on the website when I change later the screen sharing. So what we are doing now is actually we start including the omics with epigenomics. We're looking at the proteomics, transcriptomics, genome. I'm not doing microbiome yet. This is still in an early stage, and exposome as well. So we are trying to actually increase our prediction, increase our accuracy and precision for the genomic information, and it includes pharmacogenetics. In pharmacogenetics database, you only have 300 drugs. We're adding, or they are being added as this pipeline is getting more and more evidence for different drugs. Also, new drugs are being developed. This is part of the network. This is, actually was looking at a gene which is hypoxia inducible factor 1A, and I wanted to know what are the connections between different genes and which other genes I have to use, like EPAS, for example, and this gene that I have to use, VGFR, KDR, to actually make sense what I see. You can't just look at one gene, you have to connect it. There is metabolic pathways from other places. This is biases, and you have all the metabolic pathways here. There is a human. The green stuff, unfortunately, I can't increase. I had some of the slide, but the green stuff is actually the genes of my interest that are located in particular metabolic pathways. Not sure how you can see that. And reactome is over there. I highlighted the area where the genes of my interest are located. Also, you can see the yellow one, other genes that are proxy or also linked to different metabolic pathways. I will show you later on the website how it looks. So in a very short way, this is the process that bioinformatician, especially for clinical, let's say, purpose, for working with clinicians has to do. I prepare you a couple of files that will tell you how this is done from FASTQ file to VCF file, and then from VCF file to a full report, including pharmacogenetics. I will be probably, do you see the screen with the yellow lines? Anybody?
No, there is files. I can see files.
You can see file that has yellow line.
They are not opening yet.
Okay, so it means I stop sharing and I share that screen with you. So now you see?
Yes, yes, we do see.
Okay, this is, you can see it's a bash script. And it is not actually a script. it shows some few commands, but it's the length, as I said, is thousands of lines, and of G when I do the wrapper for G80K and with a proper let's say configuration. So I have to change, and I show you how does the this particular process looks. And starting new applications and changing my screen to that. So hopefully I won't mess up. So as you can see, it was done on June for one patient. So what I have is I got a raw file, and this raw file is your FASTQ file, and it looks like this. I won't go because there's too little time to explain what it is, but I can tell you that this is the line that I got. How many lines? This is a FASTQ file. Well, I can tell you the letters are just the quality, let's say, annotation, and then you have headers over here, which tells you about the machine, about the time, about the lane, about the library that was used. And later you have all the sequence particular read. And as you see, it was finished. And then we're moving into alignment. So it's a BAM file. BAM file is actually binary file or SAM file. And I use, in this case, BWA. And it looks totally different. Now you have not one line, but it's quite long, and you start seeing things that are slightly different. And I would like to give you okay, this is the part that is quite interesting because it's called cigar, right? And cigar is in a short what actually happens to your sequence, and this is your quality. And then you, of course, will have the sequence as well. So this is your BAM file that you can see. From BAM file, we need to create VCF file after many, many steps because we have to recalibrate, filter, and pick up the variants, and as well indels, which is insertion deletions. So VCF file, look how many lines I got from the original one. This is the line. And I got this line over here. Don't know how many I have to count. Very small VCF file. And this is VCF file that is already annotated by DBSNP. G80K can do it. These are the headers that you have. This is the sample name. And then you have a chromosome. Of course, you have position. You have the ID. You have reference gene, the allele. You have alternate allele. Quality, quite good. And then you have the filters and which pass, and you will see that pass the filter, and then you have the info. Info is actually all of it, and until the genotype, which will be here. The genotype, I will not to encroach to another one. Yes. This is your genotype. That's where your genotype format is declared in your VCF file. And here it is heterozygous 01 for that particular variant. And it's, there are, of course, meaning to each all of the numbers. So VCF file, which is annotated, all right? You can see like this. So this is the basic file that we should get. This is the basic file, and out of this, we can do a magic. We're going to the magic part. So I will show you my 1,000th line that, oh no, we have to go to genotyping. This was sequencing. How do we get VCF files from genotyping? Oh, that is a really pretty tough time. because the vendors don't give you a VCF file. They give you a very dirty file. This is from Illumina raw file, which actually means nothing. You don't even know what is the reference allele, what is the alternate allele. You don't even know what is the proper genotype. You don't know much about quality. Your position sometimes are not really the same, because this is the position and this is the position. So actually, when you get something like that, you need a couple of months to write your own programs to clean it up, because they report either on the bottom, on the top, or on a plus or minus, and depending on the vendor who actually submit them the variant. So when you have like this, you need to create VCF file. So what I will do for you right now, I will show you how you create a report, a full report with pharmacogenetics, having this dirty file coming in and coming out with this beautiful VCF file. You have to understand that this VCF file is developed, manufactured from that. So you don't have annotation in this VCF file anymore. I don't annotate, I don't need, because what I will do, I annotate myself, or I use it for genetic diseases in a portal that is called Genix. So what I need, what is the most important is actually my chromosome position, where ID not always, but I need the reference and the alternate allele and the genotype, which is here. So as you see, all of them at this point, because I have 400,000 out of 700,000 variant that is in Asian screening array that Illumina provides, I can annotate 400,000. So variants, quite a lot. You can use it as actually exome sequencing. And then I will have to show you the video that converts thousands of different lines and from different programs, and within less than 1 minute, and create a beautiful PDF format. So we stop sharing, and we have to go to my presentation, and this is the video. So I have to stop sharing, and where is the video, my beautiful video that you would like to see? It looks like I have to click again. Screen sharing, do you see the?
Oh, I see the full, yes. You can click on that video.
Okay, now we're going. Now I have to very quickly change, because anyway, do I see any? Yeah. Do I see anything moving here? Otherwise, I will just put you on the. Sorry, there are more windows. Is this one? Okay, I start from the beginning. I purposely slow it down to but almost 90%. Okay. Later, I show you how it runs in a full, let's say, speed. So let's go from the beginning. So this is when you see my script, which actually is a wrapper, thousands of lines, including different programs. And it will soon will show you one by one how they are being executed. And at the end, I will show you the program. This is especially slow because I want to show you how usually what it does. This is what is the result. You can relate to Illumina ASA.
and then you see what are the steps being done, and they are really difficult steps, and these are risk factors, the numbers, and you have to link it. Now, this is latex that actually is generating the report in PDF file, right? So after that, it's quite a lot. That is, if I put faster, you will see that is quite a lot. Now, what we do, we go to playback, and we go to speed that is normal speed, yes? We go to beginning, and let's go. It's less than 30 seconds, if you can count. And there are thousands of lines. And it is 8 months of work just to do it, to clean up the file and generate the report. Okay, we're done, and actually end of run. Congratulations, successfully. Now, what we need to do is to share to my application.
Excuse me, Dr.
Yes.
Suwinski. As much as we love to see this, you have 1 more minute, then we'll go to questions.
Okay, so I show you the report. Okay, and I share with you the applications. I don't want to show you everything because this is what it creates actually, all the lines. But this is your pharmacogenetics, which actually you are having so many drugs, and which one you can take, which one you don't have to take. And we go very fast to my presentation, and we move to yes, to my presentation, yes, and we move to yes. These are the content which comes from different repositories. There are primary, secondary, literature, and complex. You will read about it because it's here. And NCBI, of course, has the whole ecosystem that you can actually use. We don't have much of governing, or let's say overseeing bodies. We have to use ACMG for genetic diseases, CPIC for pharmacogenetics, and laboratory standards for let's say for tests that developed by labs. And in summary, the information is important. It tells us a lot, but it also is plastic. The genes are plastic, interacts with environment. So this is the precision medicine. We get all the information. We have the black box. This is the magician guy. And then the information comes here, being meshed with any other information about the patient. And how we look at pharmacogenetics, we look at each variant require different drug, even though it's the same disease. In US, on drugs, you have a black boxes that are telling you to do genetic tests, because otherwise you won't know whether the drug works or not. These are the processes for cancer, a different pharmacokinetics, pharmacogenetics that is using how we actually assess the effectiveness of the drug or adverse drug reactions. There is phase 1 and phase 2 of those. In phase 1, basically what we're doing, and we're trying to make the compound or the drug water soluble so it can be excreted. Liver is this doesn't actually recognize drugs as a foreign bodies, xenobiotics, so it tries to eliminate as fast as possible. So these are the genes we're looking. Remember, ethanol is always an inducer as caffeine. You use ethanol and caffeine, you're actually increasing your drug metabolism, so it means you need to take higher dosage. Also, you have to understand that food affects. For example, the biggest processor gene is, or protein is in the gut, and it eliminates 25% of drugs before they enter bloodstream. You take grape juice, you neutralize.
Excuse me,
yeah.
Dr. Suwinski, we have just run out of time. I'm sorry to interrupt you. There is just one, there is a question. If people want to ask any questions, we could move on to question session, or do we have a concluding remark perhaps from you?
I see one, trustable pipeline for variant discovery from transcriptomic data. No, I don't use transcriptomic at this point because this is research-based. In that sense, in clinical settings, we do not use transcriptomics, but I do believe GETK or MIT have for transcriptomics. And if you go to Galaxy, you will have a pipeline for transcriptomics data as well.
Okay, I believe because of in the interest of time, I'm going to ask the people who have more questions to contact Dr. Suwinski, if that's okay with you, Dr. Suwinski.
I'd be very happy. You can even do it through email, because I think you can provide email, or through you guys, and you can ask me the questions and I can answer them and then send to you. Since the time is of essence.
Thank you so much, Dr. Suwinski. This was very enlightening. I think this is a need of the hour, because the two fields, clinician and the bioinformatician, we need people like you to be the bridge between them. That's a great talk and great work. Thank you very much.
I'm sharing the presentation, so guys, you can always look what I did not mention during the presentation. Thank you very much, everybody.
Thank you
for attendance.
Thank you very much.
Patience. Okay, thank you.
Thank you so much.
Thank you.
We have our next speaker is Dr. Serdar Durda, Professor Dr. Serdar Durda. I'm going to do a brief introduction of Professor Durda. He's Hacettepe University graduate, did computational chemistry in Bilkent University, and PhD in bioinformatics and computational medicinal chemistry from Free University Berlin. And he did the postdocs in University of Calgary. And since 2013, he has been professor at Department of Biophysics, School of Medicine, Bahçeşehir University. Professor Durda is recipient of multiple awards, including Aziz Sancar Incentive Award in Health Sciences, and Respect for Science Award, and Young Scientist Award, to speak of a few. Professor Durda, the floor is yours.
Thank you very much for this nice introduction. So today, I'd like to talk about a bit the machine learning-based models and drug repurposing studies that we have conducted for the last 2 years. So before passing to our results, I'd like to just pass over mainly structure-property relationship and quantum structure-property relationships, and mainly general idea about the computer-aided drug design. So first of all, we can divide it computer-aided drug design as two main divisions. It can be direct drug design or indirect drug design. So we can say that actually, if you have the target structure, I mean the protein structure, you may go with the target-driven based design. So it is mainly docking or MD simulations, because what you will do, you will define the binder pocket, and you will try to estimate the pose of the ligands at the binder pocket. But for some of the target structures, it's not possible to get the crystal structures, or let's say the experimental 3D structure. In that case, you can go with the pseudo receptor models, such as quantum structure relationships or ligand-based models. So in our lab, we are combining all of these steps to get the models, and then define the the LAT compounds. So before passing to, as I said, before passing to our results, I'd like to just go over the background of these steps. So first of all, here I'd like to just show two structures, two small molecules. One of them is the paracetamol, as you know. It is a very well-known compound. And what the paracetamol does, it actually inhibits selectively the cyclooxygenase enzyme, COX-3, which is found in the brain and spinal cord, and consequently it relieves the pain. In the second case, we have cyanide, and the cyanide exerts its toxicity by inhibiting cytochrome C oxidase, which is the terminal enzyme at the respiratory strain, and it actually leads to insufficient utilization of oxygen and suffocation. So here, what are the differences? I mean, one of them is the relieves the pain. The second one is the just create the problem in the utilization of the oxygen. So here, the only difference is the structure of the molecules, and from this we may say property of X can be the function of the molecular structure. So from that, we can say that molecular structure may be related with the biological properties. It can be linked with the chemical properties. It can be linked with the physical properties or electronic properties and so on. Or in summary, we can say that molecular properties are coded by molecular structure. But how can we pass using the molecular structure to the property? For this, actually, we are using mainly molecular descriptors. But what are the molecular descriptors? Well, the molecular descriptors are the structural factors that influence the molecular property which will be identified, such as the molecular property can be lipophilicity, it can be steric properties, or it can be electron properties. You can increase these properties. If I want to give some examples for these descriptors, molecular descriptors can be 0D, 1D, 2D, 3D. For example, in 1D, you may have just the molecular weight or number of specific atom in this compound. In 2D structures, of course, you have more information, such as number of rings in these compounds, number of hydrogen bond donor groups, acceptor groups, or number of rotatable bonds in this descriptor. And if you pass to 3D molecular descriptor, of course, you will get more information. It will also cover 0D, 1D, or 2D informations, because you are just having all the information in the 3D, such as molecular volume, surface area, dipole moment, and so on. Specifically, you can actually go with the biological effects, such as you can use just a you can actually use a hydrophobic pocket, and you can actually get the information about the enzyme and substrate information in pseudo receptor way. So from this, we can say that if you have the enzyme and substrate, you can get the enzyme substrate complex. And from this, if we show the enzyme and E and substrate as S and the complex as ES, you can get the dissociation constant or association constant as the concentration of the complex divided by the concentration of the enzyme times concentration of the substrate. And from this, you can pass to free energy of binding. And simply, you can actually show the binding of the free energy of the ligand to the receptor as a logarithm of 1 divided by concentration of the substrate. So from this information, actually, if you just have the different ligands, and if you just measure their activities with and the lipophilicity or hydrophobicity information, you may get a linear equation. And the most widely used method for deriving QSAR equation is the linear regression, which uses the least square fitting to find the best combination of coefficients in the equation. The least squares technique can be illustrated by using a simple case where the activity of, the biological activity is a function of only one descriptor. Therefore, the form of equation will be, of course, the linear equation. And here, the activity, the y, is the dependent variable, and the x here will be independent variable, I mean the descriptors. The objective of a regression analysis is to find the coefficients m, which is the slope, and the c, which is the coefficient. And from that, you can actually try to minimize the sum of deviations of the observed values from the fitted equation. And you can get the least square coefficients m and c in this linear equation, which is given here. And here in this equation, we are actually using the mean values of the independent and the dependent variables. So usually in the QSAR, one of the mostly used technique is the free-Wilson QSAR models. In that case, you have a set of molecules there and their experimental IC50 values, or let's say biological activity values. Then you are trying to create multiple linear regression formalism. In that case, what we are trying to do, actually, we are trying to construct a structural matrix, and we search for the unknown set of coefficients b, which is multiplied by non-descriptors, which is the best approximation to the non-activity data. And it actually gives us the solution to this problem. And in that case, in order to solve the matrix, we need to go with the matrix inversion. In the structure-based methods, usually the docking is the mainly used method, and it is an energy-based operation. And what we are trying to do, we are trying to understand how the ligands actually have a conformation or biactive conformation at the binding pocket. And in order to do this, there are many different approaches. And here, what we are trying to do, we are trying to understand two things. One of them is how the bioactive conformation of the ligand is actually looks like, and the second, in this conformation, what will be the binding energy, and we are calling this docking score. And here, actually, you are seeing a general scoring function. Usually, it's a very empirical technique. What we are doing, we are trying to understand what is the binding energies are coming from the hydrogen bonds. What is the amount of interactions coming from the ionic interactions? I mean, when I say the interactions, it is the interactions between the non-bonding interactions between the ligand and the receptor, and aromatic interactions, and also together with the lipophilic interactions. And we need to also give a penalty function that because of the entropy loss here, before the docking simulations, the ligand and the receptor itself has higher entropies, and when they go to complex, actually they are losing their mobility. So for that reason, we need to also add some penalty function. So this is the basic equation for the scoring function for the ligand docking. And what kind of interactions we have? So in the docking, we need to get two things. We need to consider two things. One of them is the shape complementarity. I mean, the 3D structure of the ligand and the protein should fit to each other. But this is not enough. You should also consider the chemistry. I mean, the chemical complementarity. And this chemical complementarity can be electrostatic, hydrophobic interactions, pi-pi stacking, or aromatic interactions. Hydrogen bonds, of course, should be considered. Here, I just put one example. Usually what we are getting, you have one target, specific target with different the pose of the ligand. So here, the ligand is shown with this small orange-colored picture, and the macromolecule is of course this yellow color one. And here you see we have 7 different poses of the ligand at the binding pocket. And from this, actually, we are using in each pose, we are measuring the binding score or scoring function. And from this, we can say that the third pose actually goes the lowest delta G. So for that, you can actually use this information for next steps. So from this, you can say that, okay, this conformation of the ligand fitted well, and it goes actually, it actually brings the energy to the low state. So you can design new compounds, or you can better understand what kind of interactions you have between your ligand and the residues in the active site. But this technique, I mean, the molecular docking, is very static. I mean, you are just getting to one snapshot of your ligand and your protein. Of course, static or average structures may not be representative of the conformations, which is critical to the function. So what you need to do, you need to understand the dynamics. And for this, I mean, if you just give an example, if I want to give an example, here actually in the left figure, we are seeing a static structure of, let's say, protein and ligand. But in order to understand real conformational change during the action, you need to have the dynamics. And here, for this aim, we can apply actually classical molecular dynamic simulations. And in that case, we can use the Newton's second law. And here, the red colored ones actually are the non properties. And from this, if you just measure the force and you are also known the mass and can pass to acceleration. And from the acceleration, you can actually estimate the velocity of the second position. And if you can estimate this velocity of the second position of the atoms, you can also estimate the position, of course. And from this, if you just integrate this with the small time steps, you can actually get the how the dynamic of the structure of the protein and ligand may change during the time. So you can get time-dependent integration of the dynamics in the protein and ligand complex. You can get an idea. So so these are the basics information about what we have actually conducting during our projects in the ligand-based models and the structure-based models. And actually, we use this all these techniques to better understand the if we can identify new small molecules against the brain tumors. And here, I would like to also give some information about the gliomas, which are the common primary brain tumors. Gliomas are actually brain tumors, and they actually starting in the glial cells, and they are classified as astrocytic, oligodendroglial, oligoastrocytic, or ependymal, according to the morphological and activity characteristics of the cells under the microscope. According to World Health Organization in 2007 criteria, besides the cell morphology, also mitotic activity, microvascular proliferation, anaplasia, and necrotic activity are also evaluated as a classical classification. And in this classification, we can actually divide the tumors as from slow growing, or let's say low grade, to high grade. These are the fast growing, from grade 1 to grade 4. And we can say that gliomas are common primary brain tumors of glial origin. They are actually representing the half of the brain tumors with highly aggressive and invasive properties. Gliomas constitute the second most common cause of the death from the intracranial diseases after stroke. And they have dynamic heterogeneous tumor tissues consisting of tumor cells with uncontrolled proliferation inducing angiogenesis and escaping the host immune response. Here you may just see the low-grade glioma in the picture and glioblastoma. And we can say that gliomas actually have different type of the intratumoral heterogeneity, and they have very diverse in the tumor-associated molecular defects, which may lead to the treatment resistance and tumor recurrence triggered by the cancer stem-like cells. And the present therapeutic approach aims to target tumor drivers such as specific gene defects and oncogenic signaling pathways, but these approaches have very low efficiency, I can say. Around 50% of glioma patients are diagnosed by GBM, glioblastoma multiforme. The World Health Organization grade 4 glioma, which is the most aggressive brain tumor in adults. the difficulty in the treating gliomas is primarily due to the invasion of the tumor-containing cell populations, and despite extensive treatments, including surgical resection, radiotherapy, and chemotherapy, GBMs tend to be recurrent and fatal with a median survival of approximately 1 year. Studies show that GBMs display the worst prognosis, with only 10% of the diagnosed patients surviving only for 5 years. In the GBM, isocitrate dehydrogenase 1, IDH1 mutations are quite crucial. And IDH1 is a critical metabolic enzyme which is involved in the tricarboxylic acid cycle. And it catalyzes the oxidative carboxylation of the isocitrate acid to alpha ketoglutaric in the NADP-dependent manner by using divalent magnesium ion. And here, the key residue is the arginine 132, which is the common mutation that we see in the IDH1. And these specific mutations belong to heterozygous missense mutations and led to the naive form of IDH1 catalytic activity, which convert alpha Kg into the oncometabolite, the 2-hydroxyglutarate. So from this, actually, I will show you that initially we used cell-based line modeling, and then we passed to target-based modeling. In the cell line-based models, we used well-known cell lines in the brain tumors, especially in the GBM. But after that, we used also target-based, and in that case, we used IDH1 mutant inhibitors to construct the models. Well, for the cell line-based models, we used glioblastoma inhibitors studied against U87 and U251 glial cell lines. And here we received the IC50 values from Campbell database. And 3D structures of each ligand were prepared and used in the model. And in that case, we used the Maestro molecular modeling package to create the 3D structure of the ligands. For the first model, we used 172 molecules, and we created them, we obtained them at the neutral pH. The molecules were divided into 2 groups. 67% of the molecules is used in the training set, and 33% of the models are used in the internal validation set. For model generation, 116 compounds were divided into 70% of the training set and the 30% of the test sets. And of course, we want to also use a set of compounds to validate our models by external test set. When I say external test set, this means that these molecules, their activities, their IC50 values are already known, but these molecules are not used in the construction of the model. So after the construction of the model, we ask the model to estimate the binding affinities, or let's say IC50 values of these compounds. And based on this prediction, we can actually validate if our models are able to predict correctly the unknown compounds activities. So for the deep learning-based machine learning models, we actually constructed and we tested quite different set of approaches. And finally, we found that KPLS-based model gave us the successful results, and its R-square values was 0.89 and Q-square values was 0.80. I mean, statistically, it looks okay, but this graph actually shows the observed versus predicted PIC50 values of the known compounds with the internal test set and internal training set. And then we just used 169 compounds in the external test set to see if this model, constructed models, is able to predict unknown compounds activities. And in that case, the R-square value was 0.53, which is good. I mean, if you have more than 0.5, this means that your models may predict relatively in a good power of the predicted activity. A similar approach is also conducted for another cell line, which is quite important in the brain tumors, which is the U251 cell line, which we called model set 2. The previous one was constructed with U87. So this one is the U87, and this one that I will describe is the U251 cell line. In the model set 2, we used 144 inhibitors, and we divided again these molecules to training set and the test set. And we also used 56 molecules as the external test set. Here you may see the scores. The R-square value in that case is 0.90, and Q-square value is around, again, 0.90. So this model that's actually created with U251 statistically has improved a bit more. And again, we used KPLS model, and I mean, we found that within the used approach, the KPLS was one of the most successful approach to construct the model. Here you may see the predicted and observed PRC50 values of the training set and the test set molecules. And here you see the, again, the external test set for the model set 2, which is the U251 cell line inhibitors. And here we really see quite improved R-square value. I mean, previously it was in the U87 cell line models, it was 0.53, and now it is actually increased to 0.79. So this means when we create the model with the U251, we are getting much better estimation, much better predicted values for the constructed models. We also try to see if we can get a similar trend with the 3D QSAR-based models. And in that case, we used phase 3D QSAR model program. And we get two different hypotheses. One of them is the six-sided model. Each letter here shows different pharmacophores, such as R shows aromatic rings, D shows hydrogen bond donor groups, A shows hydrogen bond acceptor group, and H shows hydrophobic groups. And here we may see these groups in one of the active molecules. So here, for example, we have 3 R groups, three rings, 1 hydrogen bond acceptor group, 1 hydrogen bond donor group, and 1 hydrophobic group. So we constructed quite hundreds of the different hypotheses, but these two hypotheses, 5-sided AAA, 3A, and 2H, and 6-sided 3R, 1A, 1D, 1H hypothesis gave this successful results. You can see the R-square and Q-square values, which are more than 0.70. And here you can see the predicted and observed values of the training set and the test set. Again, we try to see if this model constructed by the phase classical 3D QSAR will give us the successful external test set validation. When we say it, for example, in the 6-site model, at least 3 sites must match to the model. In that case, we received the total number of compounds, of course, will be decreased because in the external test set, it may not have these sites. So, but the R-square value was low. I mean, it was 0.17, which is not useful. When we pass to 4-sided, when we say that 4 sites must match, in that case, the total number of compounds is decreased more, and the R-square value is 0.39. And when we pass to 5-sites model, 5 sites must match the model. In that case, total number of compounds decreased, but the R-square value was more than 0.60. Along with ligand-based pharmacophore models, we also constructed hybrid, the ligand and structure-based models. In that case, what we did, we used 9 non-IDH1 mutant complexes from the protein data bank, and we constructed our pharmacophore models at the binding pocket. And in that case, we received, again, 6-sided, 5-sided, and 7-sided models. And here you can see how these models looks like. Then, after construction of the models, we conducted a virtual drug repurposing study. And in that case, we used 2,367 FDA-approved compounds from the drug bank. And we conducted structure-based and the ligand-based techniques. When I say the ligand-based, we used the constructed models, such as deep learning-based machine learning models. We also used the QSAR-based models. And when I say structure-based models, we just docked these molecules at the two different binding pockets. And here you can see the binding cavities, which have, since it is dimer, we have two different binding pockets. So after that, after getting the ligands with the high docking score and high predicted PI security values, we selected these hits and we run first docking and then MD simulations, and then we perform MMGBSA binding free energy calculations. So here you may just see for each model how many compounds actually we received with our SATET cutoff values. For example, in the KPLS radial 15 model that we constructed, we only received one compound in the 542 binding pocket and no compound in the 544 model, because none of the compounds actually passed our cutoff value. And we may see the other, the total number of other screening in our models. In the MD simulations, after running the short MD simulations, first we run 10 nanosecond MD simulations, and for this 10 nanosecond MD simulations, we used all of the compounds, which is actually passing all these models. For example, here in this model, we have 31 compounds, so we use these 31 compounds in docking, and then we use top docking pose and we run the short MD simulations, I mean the 10 nanosecond MD simulations, and based on the probability density of the MMGBSA coming from the short MD simulations, we selected this cutoff. I mean, we used this cutoff, and we just performed the long MD simulations for these molecules. And after getting the long MD simulations, again, we conducted MMGBSA to get the average binding free energy calculations. And based on this, we selected hit compounds. And at the end, we received 4 compounds, and we run 500 nanosecond MD simulations for very long MD simulations for these 4 top hit compounds. You can see here this 4 top hit compounds that we selected are decualinium, conivaptan, tazemostat, and terotristat ethyl. And from these 4, among these 4, decualinium was the best compound. In order to understand which residues at the binding pocket is more crucial to the binding pocket, we also went to the per residue interaction analysis, and we calculated the binding energy contribution coming from each residue. And from this, we can say that these 3 residues, tryptophan 124, tryptophan 267, tyrosine 285, is the most active, and actually they contribute more to the binding energies.
Excuse me, Professor Duda, you have a couple of minutes for.
Okay, yeah, I just finished.
Please wrap up. Thank you.
Yeah. So here we may see the 2D and 3D representation of these compounds at the binding pockets. Here in the top, you can see the reference molecule and their interactions, such as in the reference molecule, you can see that tryptophan 124 is crucial, isoleucine 228 is crucial, and actually our selected molecules also have these similar interactions. We actually repeated similar things, but in that case, we used, instead of using the cell lines, we used the target. In the target, we use IDH1 mutants. Initially, we constructed 100, we actually received 197 molecules from the Campbell, which were IDH1 mutant inhibitors. And we also used another 85 compounds as test set, external test set, to validate our models. And here you can see the R-square value of the external test set, which is 0.51. And we can use this model in the screening. We also use canvas neural networks to construct the models. And in that case, we used PRC50 values as a Y variable to make the predictions, and the physical chemical properties were used as X variables to train the neural networks for predictions. And we used 77% of the learning set was divided into training set, and 3% was used as internal test set. And here you can see that when we use the neural network, the R-square value of the external test set was increased to 0.56, which is good. We also used binary fingerprints from the KPLS regression to create another model, and in that case, the R-square value was 0.54. And we used this model to screen again the molecules in the FDA-approved compounds. And docking scores showed that pentagastrine, for example, was one of the best compounds, which together with the docking score and in the constructed models, it actually gave us the better scores. Still, our simulations, MD simulations for the second part is continued. So unfortunately, I cannot give the details of the MD simulation results. We are still waiting the results. So when we get these results, we will also try to combine the first part, and we will run in vitro results. We will, of course, these molecules are ready, so we will order them and we will test them using the experimental methods. So, in conclusion, we can say that results from U251 actually gave us the better successful results compared to U87 cell line. Machine learning-based models compared to 3D QSAR gave better successful results. And overall, within the scope of this project, NIF compounds, more than 10 compounds actually, were identified as hit compounds, and among them, selected compounds are ordered for in vitro tests. Okay, so this research is actually supported by TÜBİTAK. It's a Turkey, Korea bilateral call. So this project is actually supported by Turkish Research Council, and also our university supported our project. Two of my students, Mihrin and Farid, actually work on this project. Thank you very much.
Thank you, Professor Durga. There are a couple of questions from the chat I can see. Someone is asking how many replicates of MD simulation are needed to generate the box plot of the MMGBSA for each drug candidate? That's light, high, long.
So here actually we repeated our simulations by 3 times. So 500 nanosecond times 3 for each complex.
And the same person is asking, is it fine to not set any seed value when conducting MD simulation?
Well, we are usually using the different seed numbers to get the different average velocity in the beginning, actually.
Any other questions? If there are no other questions, I would like to thank
Martin,
Professor Durga. Yes.
There's one question I'm not sure if you already read. Is it fine to not set any seed value when conducting MD simulations to compare the protein interaction with different peptides or ligands? This is from Lai also.
Yeah. This is I try to explain. For the replicate, actually, I set different seeds, but if one needs to try to get the if the protein ligand will give you the similar, or let's say the same pattern, you may of course set the same seed number. But here in our case, I used different seed numbers.
Thank you very much, Professor Duda.
Thank you. I hope this background information and also the information I gave about our research may lead new ideas of the audience for better therapeutics in the brain tumors or in their specific fields. Thank you very much.
Thank you. Our next speaker for this session is Dr. Tunahan Cakir. Dr. Tunahan Cakir is Associate Professor of Computational Systems Biology at Gebze Technical University. He did a postdoc in biosystems data analysis at University of Amsterdam and University Medical Center Utrecht. He has also received multiple awards, including the Young Scientist Outstanding Achievement Award from the Turkish Academy of Sciences and the Research Incentive Award by Professor Mustafa Parlar Foundation. The title of his talk is Transcriptome Mapping on Cellular Networks to Elucidate Molecular Mechanisms in Cancer Dormancy. Professor Tunahan Cakir, the floor is yours.
Thank you. So you can hear me well, right? No problem.
Yes.
Okay, good. Thank you very much for the introduction. And I also thank the organizers for giving me the opportunity to share our recent results of cancer dormancy. So my talk is about transcriptome mapping on cellular networks to elucidate molecular mechanisms in cancer dormancy. Let's look at some statistics first. Each year, about 20 million new cancer cases are reported. And if you look at the number of deaths from cancer, it is almost half of this number, 10 million, almost 10 million per year. And risk of dying from cancer before age 75 is 20%. So we have a deadly disease, and the major reason why cancer is so deadly is because of its metastatic behavior. So cells can leave the primary tumor and they can use the circulation system to go to other organs, and they are located there and they can start proliferating there, and this is what we call metastasis. And the major problem here is what we call cancer dormancy. Normally, cancer cells, they proliferate, and the standard treatment alternative in cancer, which is chemotherapy, they target proliferating cells. When cancer cells go to distant organs, they can stay in dormant state. So this means that they do not proliferate, but they are still alive. So they are kind of sleeping. And since they do not proliferate, they escape from standard way of treatment, which is chemotherapy. So you think that the cancer is treated, but you are never sure that there are dormant cells in the body of cancer patients. And you never know when they will awake. So sometimes it takes a couple of months, sometimes it takes years, and then they are awakened for some unknown reasons, and then the metastasis leads to the death of the patients. If you look at the molecular features of cancer cells, you can look at the genome. That's one of the alternatives. And genome gives us static information. I always give this example of butterflies versus caterpillars. They have exactly the same genome, but if you look at the functional genomic information, you will get more information about why they are different. Genome doesn't tell us anything about why we see this different phenotype, but the data sets that are called as functional genomics data, such as transcriptome, proteome, or metabolome, they tell us more about disease effects and the molecular phenotypes. In our research, we have focused on transcriptome because it is, in terms of the cost, it is in lower cost compared to the other functional genomic data such as proteomics and metabolomics. And it has a wider coverage of the genomic information. Let me go through the relations between genomes and transcriptomes and some other molecular information. Then I will go on with our research. So some part of genome is active. So genome is the set of genes in the organism. Only some genes are active in a given condition, and we can track this by using transcriptomic data. And mRNAs that make up transcriptomic data are used to make proteins, and proteins are not isolated from each other. They interact with each other, making protein interaction networks. You can also measure protein levels that make proteomics data sets. But as I said, transcriptome data sets, they are cheaper to get and they have much wider genomic coverage. So in our research, we have focused on transcriptome data. One other molecular interaction network relevant here is that some proteins known as transcription factors, they go and bind to the promoter regions of their target genes, leading to gene regulatory networks by switching on and off the target genes. So in this first part of my talk, I will share with you how we have integrated cancer dormancy specific transcriptome data with protein interaction networks and gene regulatory networks to identify dormancy specific subnetworks. This has recently been published in scientific reports. The transcriptional landscape of cellular networks reveal interactions driving the dormancy mechanisms in cancer. The research is in collaboration with Devrim Gözüaçık from Koç University, and the research is supported by a TÜBİTAK grant, and actually Devrim Hoca is the coordinator of the project. And my student at the time, she was a master's student, and this was her master's thesis topic. She performed almost all the analysis regarding this work, Dilara. So first, we went to literature to identify dormancy-related transcriptomic datasets. This is a relatively very new field. So although cancer dormancy was known for the last decade, transcriptome-based analysis of cancer dormancy is a recent research of, let's say, last 5 years. So if you look at the published the year they were published, it is mostly the last 5 or 6 years. So when we went to literature, we found cancer dormancy comparisons for a number of cancers and a number of datasets. We focused on 10 transcriptomic datasets, which allowed us to compare 21 different cases. So in one dataset, there can be more than one different cell lines. So they have made dormancy models in vitro conditions by using cell lines, by using multiple cell lines. So that's why number of compared conditions is more than number of datasets. So we use this transcriptome data to map this data actually on protein-protein interaction network of human. This was obtained from literature, a protein-protein interaction network that included about 17,000 proteins and 300,000 interactions were used in this study. For gene regulatory network, again from literature, we compiled the known molecular interactions, which included about 6,000 genes targeted by about 1,000 transcription factors, and the number of interactions between those transcription factors and targeted genes were about 20,000. For each of those 21 cancer dormancy comparisons, we used transcriptomic data to identify, to calculate p-values of about 20,000 mRNAs in the data. Then these p-values were mapped on protein-protein interaction network and gene regulatory network separately to identify a dormancy interaction network. For mapping, we use two well-known algorithms in literature. We have used one additional network in the analysis in addition to protein-protein interaction network and gene regulatory network. This is actually a modification of gene regulatory network, which we called transcription factor-free gene regulatory network. So a transcription factor targets genes. So a transcription factor can target multiple genes. In this TF-free network, we removed transcription factors from the network, and all the genes that are targeted by the same transcription factor were connected to each other to make a TF-free network. And this led to a network of about 6,000 genes, so no transcription factors in this network, and about 2.5 million interactions. So in summary, we used 21 dormancy cancer comparisons. We used 3 interaction networks: protein-protein interaction, gene regulatory interaction, and TF-free gene regulatory interaction. And we used 2 mapping algorithms, APBioNet and key pathway miner, leading to, in total, 126 subnetworks. By subnetwork, I mean a network that consists of significantly changed genes in the cancer dormancy comparison, and also physically interacting with each other or interacting based on gene regulatory interactions. And the size of those subnetworks were about 100 to 200 genes or proteins. So we fed about 17,000 genes or proteins in the case of protein-protein interaction network to the algorithm, and we found the genes or proteins that are really significantly affected and also physically interacting with each other, and the number is much lower compared to the input. So these networks included about 100 or 200 genes or proteins. Then we used majority voting. So of those 126 subnetworks, which genes or proteins come up mostly if we score them. And we focused on upregulated genes because in the project, the overall goal was also to identify some drug targets, and most of the drugs, they suppress the targets. So we wanted to identify the genes, the proteins actually, they have physical interaction or gene transcription factor interaction. And then those genes are upregulated in dormancy, so you can use potential drugs to suppress them. And in this way, you can identify dormant cells in the body. You can suppress them, they can escape from chemotherapy, but with novel drugs, they can be targeted. So at the end of this majority voting, we identified a network which we called dormancy interaction network, which included about 140 genes and about 200 interactions. Some of those genes were identified by all 3 network approaches: protein interaction networks, gene regulatory interactions, and also TF-free gene regulatory interactions. Some of them were only identified by protein interaction networks, but we consider those different networks as complementary to each other. So some dormancy-related genes can escape if you just focus on one approach, or one bioinformatic algorithm. So by using those multiple networks and multiple bioinformatic tools, we wanted to get a more thorough list of dormancy associated interactions. Of those 139 genes, when we go to literature, so these are all independent research research sources, we have found 15 genes. They were reported to be upregulated in cancer dormancy. For example, CDKN1A, it was reported to be induced in dormancy, or THPS1, again, reported to be induced in dormancy in breast cancer and glioblastoma. So those sources, they were not used in our algorithm, but we were able to catch those already known dormancy proteins. They were available in our network. As I said, dormancy and study of dormancy by using omics approach are still not very common. So it was not that surprising to identify a lot of genes or proteins that were not yet reported in literature. So as a quick summary, we have identified a consensus dormancy cancer interaction network from these 21 cancer dormancy transcriptomic comparison. And several genes in the network were linked to dormancy or metastasis. And the nodes that were not reported to be dormancy associated, they are actually candidate genes for further studies, for the treatment of dormancy. And actually, some of them, they were not directly associated with dormancy in literature, but a group of them, as I said, they are mentioned in metastasis-related research. So this was the first part of my talk, which focused on integrative analysis of transcriptome, dormancy cancer transcriptome with protein interaction networks and gene regulatory networks to identify dormancy-specific interaction network. In the second part, we did the same mapping, but this time we mapped dormancy transcriptome data on genome scale metabolic networks to identify dormancy-associated metabolic pathways. This is a work under review. Again, a great collaboration with Devrim Hoca from Koç University. And this work was master thesis of my student, Merve. So transcriptome mapping on genome scale metabolic networks highlights metabolic signatures of cancer dormancy. Let me emphasize again, dormancy is a very recent research topic when it comes to molecular players in cancer dormancy. So there wasn't much sources that talks about which metabolic pathways are altered or dysregulated in cancer dormancy. So we used the same approach, I mean, the transcriptome-based approach, to identify metabolic pathways that were altered in cancer dormancy. Why metabolism-based approach? Because cancer is a metabolic disease. The most well-known feature of cancer cells is Warburg effect, which is a metabolic alteration. So normally, normal cells, they have an active TCA cycle, active oxidative phosphorylation, and they provide, they make their energy, ATP, by using oxidative phosphorylation, by using mitochondria. And therefore, they have a high rate of oxygen uptake compared to their glucose uptakes. What we see in cancer cells is a much higher glucose uptake and surprisingly inactive mitochondria, almost inactive mitochondria. So rather than using mitochondria as their energy source, they use glycolysis to make their ATP. This is what is known as Warburg effect. And because of this highly active glycolysis, they have a high rate of lactate production. So this is the major specific major properties of cancer cells. Cancer is a metabolic disease. That's why it was relevant to identify what kind of alterations were there in cancer dormancy. So just remind this figure that I have shown you. Some proteins, they are enzymes, which means that they catalyze the conversion of small molecules to each other in the cell metabolism. So glucose comes inside, and glucose is a small molecule. It is converted to a number of other small molecules, such as amino acids, nucleic acids, etc., or lipid molecules, fatty acids, by using those enzymes. And those small molecule network is called metabolic network. So in this study, we have mapped transcriptome data on metabolic networks. What is a genome scale metabolic network? If you look at DNA, so there are genes, and some genes, they code for enzymes. So those enzymes catalyze those metabolic conversions. Genome scale metabolic network is a metabolic network where for each reaction, the corresponding gene information and enzyme information is also available. So you can use those networks for integration with transcriptomic data. So as transcriptomic data set in this study, we have used two different transcriptomic data sets that allowed 8 cancer dormancy comparisons. One was colorectal cancer that included 6 cell lines. And the other one is leukemia, which included a bulk cancer data and also single cell data. So 3 cases, 8 cancer dormancy comparisons. As human genome scale metabolic network, we used RECON2 reconstruction, which included about 7,000 reactions, about 2,000 genes, and 100 metabolic pathways. And we used initially a mass balance-based approach. I won't go into details of those. So we used an algorithm called GMA for structure prediction. How does this structure change in dormancy, and also an approach called flux balance analysis to predict metabolic reaction rate, metabolic fluxes. So in the transcriptome data, some genes have high expression, some genes have low expression. Basically, in this approach, we use this information. So if a gene is lowly expressed, we assume that this gene is not active. So the corresponding reaction in the metabolic network will not be active, and you will have a tailored metabolic network. In the second approach we use, the approach is as follows, which is known as reported pathway analysis. Let's say you have a pathway of 3 reactions. Those pathways, they will have associated genes that catalyze, that code for the enzymes that catalyze those reactions. And if you have cancer dormancy transcriptome data, you can identify p-values for those genes. And so this is the pathway, the sketch of the pathway here. In reported pathway analysis, first, the metabolites in the network are scored based on the p-values of the neighboring reactions. In this case, p-values of G1, G3, and G2. They are used to calculate a p-value for the metabolites. And this is repeated for all the metabolites. So now you have p-values for the metabolites in the pathway. And then those p-values of metabolites are used to calculate a p-value for the pathway. In this state, you also include the reactions to the picture that traditionally are considered as members of other pathways, but they actually affect the current pathway by consuming or producing the metabolites in this pathway. So we have 8 cancer dormancy comparisons, 3 bioinformatic approach, mass balance-based approach for flux prediction and structure prediction, and a graph theoretical approach for pathway prediction. This led to 24 different results. So before going through the results, as I said, there was really little information about metabolic alterations in cancer dormancy. So what we have found is, though in dormant cells, normally in cancer cells, we have Warburg effect, oxidative phosphorylation is almost inactive, but in dormant cells, there's a switch to oxidative phosphorylation. And in relation to this, there is less lactate production compared to the cancer case. And again, compared to the cancer case, glucose consumption rate is lower, but compared to the healthy case, those cells have higher glucose consumption. And a couple of pathways were reported to be dysregulated in cancer dormancy. These were urea cycle, lipid pathways, and reactive oxygen species. When we looked at our reported pathway analysis results of 3 major cases, so colorectal case and the leukemia case of bulk data and single cell data, we have identified oxidative phosphorylation, citric acid cycle, TCA cycle, urea cycle in our results, also reactive oxygen species. I won't go into details of the results too much, not to take too much of your time. When we use mass balance based results, so the structure prediction and flux based prediction, urea cycle was there again. And lipid associated pathways reported in literature, sphingolipids, glycerophospholipids, and fatty acids, they were not identified in the reported pathway based approach, but they popped up in mass balance based approach. So these are actually the bioinformatic algorithms based on different principles. So we have seen that they are actually complementary to each other to capture dormancy associated alterations in metabolic pathways in a complementary way. So with this approach, we were able to fill in a gap in the literature. So there was a comprehensive study that reports alterations, metabolic alterations in cancer dormancy. So we have provided a comprehensive catalog of cancer dormancy related metabolic pathways. So several pathways, they were not reported in literature, but as I said, there were not that many studies in literature that focused on metabolic alterations in cancer dormancy. So those are candidate pathways researchers can focus on in their research if they are interested in cancer dormancy. That said, I will end my talk here again by thanking my collaborators. So all those research, they were part of the TÜBİTAK grant coordinated by Devrim Gözeçik from Koç University. His students contributed to research. Pınar is my collaborator from my university, my department, and Merve and Dilara, they did all the works actually. And actually in this grant has also experimental part. So in Devrim Hoca's lab, they have made in vitro cancer dormancy models. And from those models, we have collected multi-omic data, RNA sequencing data, microRNA sequencing data, proteomic data, metabolome data. We are working on using those multi-omic data sets to get a better understanding of cancer dormancy. And the related publications are in the phase of writing, and the results will be shared, I think, next year with the scientific community. I thank again for listening and for giving me this opportunity to share our results with you. Thank you.
Thank you very much, Professor Cakir. There is one question in the chat, which is, what is the by Bushra Aydin? She's asking, what is the logic behind selection of dormancy associated upregulated genes, since downregulation and suppression might be driver of the dormancy?
Exactly. So of course, I don't say that downregulation is not important. Our overall goal there was to identify to use this cancer dormancy interaction network for to identify drug targets. So since drugs target most of the time, most of the drugs, they suppress their targets, we wanted to identify a network which included upregulated genes. This doesn't mean that downregulation is not important in cancer, in dormancy, of course.
Thank you very much again. And also thank you very much for keeping the time limit. That was very good. I'm going to wait for more questions. If anybody wants to ask questions through voice.
Can I ask?
Please go ahead.
Sure,
Okay.
sure. Ask. Yeah.
Hi, Tunahan. Thank you
Hi.
for the presentation. Asif here. Quick question. Really nice to see that you are investigating these subnetworks for cancer dormancy. I'm not sure, maybe you did touch upon this as I was going in and out. Do you see these any commonality between different cancers with regards to these subnetworks, or they are kind of unique signatures for different subcancers? Maybe there are few that are shared. Could you comment on that?
Yeah, very good question. Thank you. We didn't specifically go into that because of the limited number of data sets. But if you go to literature now, even the last year and this year, new data sets are available. This is the topic of future research. So we wanted to get since this was lacking in literature, a generic dormancy signature network. But one thing, for example, we slightly looked at is the difference in the solid tumor, solid cancer associated genes, and also liquid cancer associated genes. Yes, we see some differences. As I said, we didn't go to the details of this. They are not totally separate. So if we look at the intersection, again, most of the identified genes are in the intersection, but there are a couple of genes consistently appearing in the solid tumors or liquid tumors. As the number of data sets increase, of course, such studies should be performed for cancer type specific, in a cancer type specific way. Yeah, you are very right.
Thank you. And if I may just add on, did you find, with regards to those that you identified within the network, in terms of functional role, like ontology-based analysis, I'm not sure if you covered this, do you see any particular congregation in for particular functions, preference of some functions over the others?
Yeah, good. That was one thing I didn't put into the slides. Yeah, of course, we applied gene enrichment and pathway enrichment analysis to identify what are common in those, common for those genes. There wasn't any, most of the terms we identified, they were cancer associated, the pathways that we already knew, for example, cell cycle. We know that, those cells, they don't divide. So it's not surprising to identify cell cycle related terms as a commonality. And there were a couple of cancer associated pathways identified.
Thank you.
There are a couple more questions on the chat. Ishaq Nuren is asking, hi, Professor Cakir, interesting and wonderful talk. May I know how genome scale metabolics network helps you in elucidating to protein network interaction?
So these are two different things and two different studies. The first part of my talk was on protein interaction network and gene regulatory network based analysis of transcriptomic data. The second part was genome scale metabolic networks, independent studies. There are in literature some studies on how to connect those networks, but in this research we did two separate studies. They were not linked to each other.
Another question by Anuj Mavlankar. He's asking, nice talk, Dr. Cakir. Just wanted to ask, does the data set you considered for the study have similar experimental conditions, and what comparisons have you considered?
Yeah, so we tried to include almost all dormancy cancer associated data sets in our study. As I said, there wasn't much. We were able to find 10 data sets and 21 comparisons in total. These are all cell line based studies, and there were, of course, very different approach in turning your cell line to dormancy state, to switch the cell line to dormancy state. Since the number of datasets is not that big, we couldn't group them based on how they made dormancy models. As I said, just to fill the gap in literature, we included all those transcriptome datasets that we could find in literature, regardless of how the experimental approach was to make the model, dormancy model.
Great. Could you stop sharing screen, Professor Cakir? It might actually allow for better interaction.
Yeah, sure.
There is another question. He's asking, Johnny is asking, what challenges did you face in this study? And probably, what is the main challenge?
I mean, these are the approach we already use now in my research group in general, the bioinformatics parts, and we are already familiar with processing literature-based transcriptomic data. So there wasn't much challenges. Maybe one challenge was how to combine all those results, because we obtained so many different results by using different algorithms, by using different networks, etc. Different, as I said, bioinformatics algorithms. So one challenge was how to combine all those results to get one final cancer dormancy interaction network, or one final list of affected metabolic pathways. So we use different scoring algorithms in that. I didn't go into details in my talk. So these are available in the papers.
Thank you. I'm waiting for more questions. If there are no more questions, I would like to thank Professor Dr. Tunahan Cakir and also the other speakers of the session. This was a great and enlightening session. Thank you everyone for your patience, and sorry about timing earlier, but I think we compensated later. Thanks to Professor Tunahan Cakir. Thank you everyone.
Thank you, Dr. Metin, and our honorable keynote speakers. I hope everyone here has enjoyed their talks. It's time for a small break. After the break, we will be back at 11:30 a.m. Turkish Standard Time to begin our parallel sessions. So before we let you go, I would like to convey some housekeeping messages that we will have two parallel sessions, one for CADD SARS-CoV-2, and the other one is for BioTools big data analysis and general bioinformatics. So please join your room of interest via breakout tab in your lower right on your Zoom screen. See you in 10 minutes. Thank you. Welcome back, and thank you for joining us once again. We begin our first oral and poster presentation session. We will have two parallel sessions, one for CADD SARS-CoV-2 and the other one for big data analysis, biotools, and general bioinformatics. Please join your desired rooms from breakout room tab in the override on your Zoom.
What's happening, Nihan?
Well, something is going not correctly. I just wanted to have a room with just Professor Yelekçi and Dr. Onur. I'm trying to do that, so just the 3-4 of us.
I think one of the hosts is closed the rooms mistakenly.
Give us a minute just to see what's going on. Okay, I think we found the problem. Apologies for that.
Dear all, now we closed all other rooms. Now we are going to continue BioTools presentations in the main room. Thank you.
So we will keep going with the presentations for BioTools.
Similarly across genus and across family as well. As you can see over here, we also mentioned the amount of species per group. For instance, in our order hierarchy, we get 428 average species, family, for instance, 194. IMD performed significantly better. As you can see, we get an average of about 90% in ORC. Specificity is a bit low, but our sensitivity is a bit higher. And since ORC gives the overall picture, it seems like our scores seem to be quite good. And as you can see, we have about 291 species for order and family and genus. And colorectal cancer does not perform as good as IBD, but we note that our ORC significantly seems to be quite good. And similarly, we found the same trend across the different number of groups. Also, about similar amount of species per group. As you can see, family has approximately about 100 species. And we also find the most significant group, as I mentioned before, the K groups. And then you can see over here that we have 36 species that are shared between each of the 3 groups. This could provide a potential biomarker for us, showing that since it's shared between all 3 groups, that it has higher chances of being a potential biomarker. Similarly, for each of our groups, we use something called robust rank aggregate, which is make sure that our rank that we obtain is robust. And it's a p-value of less than 0.05. And we found that we have 716 species from our top K order groups, similarly 190 from family and 103 species from genus. Moving on to the multiple feature selection using multiple classifiers here, this is a classical approach where we use different multiple feature selection techniques: information gain, MRM, select K-based, fast correlation-based filter, XGB, and information gain. We filter test it with these different types of feature selection techniques. And then we train it on different types of classifiers: random forest, adder boost, decision tree, support vector machine. We then use stacking classifiers as well, using support vector machine and KNN. And we also use stacking classifier with logistic boost and KNN. And then we compare the results with according to microBioNet, and we compare them against the top three combination of classifier as well as feature selection technique. As you can see, we use SKB with RF. Random forest, we use XGB with adder boost and SKB with logistic boost. As you can see over here, these are the top 3 combinations of feature selection as well as classifier. And as you can see, our microbiome seems to perform quite as good as similarly to the top 3 performing classifiers. As you can see, we have a very comprehensive metric, and we notice that our results seems to be quite similar. And then you can see in type 2 diabetes, our specificity seems to be quite higher as well, and our accuracy seems to be quite similar with using standard deviation, but our recall seems to be a bit lower in type 2 diabetes. And yes, and finally, for our future studies, we're thinking of grouping on different biological domain data. For instance, I mentioned before that we are planning to use gene expression data, so we are planning to use gene expression data with biological domain data. For instance, we can include click pathways, or we can also include gene ontology pathways as a different type of grouping factor, or maybe the different types of diseases that are mainly known for different types of bacteria as another grouping factor. And finally, we're thinking of grouping all different types of biological domain data as a multi-omics approach in microbiome studies, which will be part of our next study. Finally, thank you all for your keen attention. I would like to thank my co-advisor, Burjo Hoja, for her advice, as well as Malik Hoja for his support and advice throughout my whole project. You can contact me at my email, as mentioned over here, and finally my LinkedIn. Thank you very much. Hope you have a good day.
Thank you, Amar. Thank you for the presentation. Quite elaborate, and seems like you've done a lot of work. So I am, we are ready to take questions for Amar. Any questions for him?
Amar, did you use any regression model or microbiome wide association technique to correct your p-values?
Amar, are you here? Oh, you cannot speak. Nihan, can you help unmute him? Just hang on for a bit.
Just a second.
Yes, Professor, could you repeat your question if you don't mind?
Sorry?
Could you repeat your question?
Okay, did you use any regression model, or did you use any microbiome-wide association for correcting your p-values?
Our p-values was based on the ranking. For instance, we get different ranks because we used 100 cross-fold validation. So we get different ranks of each fold. So based on that, we used something called the robust rank aggregator. This, based on the ranks that we get from each fold, it gives a p-value based on our ranking. So that shows how robust our ranking measure is. Because we have noticed in our previous studies that they mention if we use only frequency, it is not used as a robust measure. So instead, we use robust rank aggregate. But regarding your question, we did not use any other microbiotes. We just only used profiling data.
Okay.
Amar, I have a question.
Yes.
How? I mean, just for the benefit of the audience, we have tools like Quine or Quine and Mothur. So I think people use that for this taxonomical, studying the human microbiome. So, I mean, when should they come to your tool? How is your tool different from the others? I'm sure yours is a bit more specific than the others. Could you just enlighten us in terms of the unique selling point of MicrobioNet?
One of the unique selling point I would say is our focus on the grouping factor. Our grouping factor, for instance, if in a clinical setting we want to find a specific genera for a specific disease, we can give you the top ranked genera that are related to this disease. For instance, in type 2 diabetes, you can get, for instance, I don't think I can share my screen, but I will just explain onwards. For instance, we find this top genera for type 2 diabetes, which shows highly significant change between healthy and disease individuals. So for that reason, we can use our tool with the top ranked genera, and we can find out, we can focus on these types of genera to find different types of applications in the clinical setting.
Okay. All right.
I have a question if everybody is done.
Yeah, go ahead.
Hello, hi, good work. I'm just curious about the computational resources that is needed to run an analysis with your tool. And I think I might have missed that part if you even have mentioned. And another thing,
why would I go to biomarkers instead of sequence similarity approach? I mean, biomarkers, I know that it's, as I know, there are not much biomarkers, but we have lots of sequences in the databases that we can build the database and then do a sequence similarity search. Does that make sense?
Yes, I think I understand your question. Regarding the computational approaches, it was used on a laptop. So it was, I think, a Core i5 10th gen Intel Core, and it was 16 GB of RAM. And it ran for, I think it was about a day, if I'm not mistaken. And regarding your sequencing, like I mentioned, our tool is focusing on the grouping factor. So for instance, if you wanted to find sequencing data, yes, you are right. You can get sequencing information correlation true. But our tool is finding on the top groups. This would help to lower the cost of find. For instance, as you know, the computational approaches using sequencing tools are quite high. But this instead can be using clinical approaches. For instance, if you want to find a specific species, and we know that it's show high correlation with the disease, we can instead use our tool with low computational cost, thereby giving us easier approaches for different types of clinical settings. For instance, we want to find whether these bacteria are shown in this disease. So instead of using different types of other measures, you can use the top ranking species to show that this disease is there in a patient, for instance. I hope I resolved your questions.
Yep. As I understood, you are using a binning approach where they use in the assembly in the genome, but you are doing it with the biomarkers.
Something like that, yes.
Yeah. All right.
Thank you. Thank you, Amar. Thank you for all the questions. We move on to the next speaker. It's a poster presentation entitled Integrated Bioinformatics Analysis Reveals Key Genes and Pathways in Lung Cancer by Mohita Mahajan and Sukanta Mondal. Nihan, please go ahead.
Hello everyone, this is Mohita Mahajan, a PhD student from the Pilani Goa campus. So today I'm here to present my work titled as Integrated Bioinformatics Analysis Reveals Key Genes and Pathways in Lung Cancer. So cancer is the second most common reason for the death in the world, and lung cancer is the leading cause of cancer death. So here we have analyzed the microarray data to identify the key genes and pathway in lung cancer. So this is the workflow that we have used in our study. So first we collected the data from IMPLIED database. Then we screened out the genes which are differentially expressed from the selected data. And then we carried out the functional and pathway enrichment analysis for the selected EEGs. And then we performed the PPI model and Harkin analysis. And after that, we have done the survival analysis for the selected genes and then validated their expression. And then we constructed the mRNA, microRNA, and transcription factor network. Now coming to the results of our study. So these are the 4 data sets that we have analyzed. And these are the Venn diagram which are showing the common DEPs among these 4 data sets, and these 952 genes we have used for further analysis. So then after identifying these DEPs, we wanted to see in which biological pathway and process they are involved. So we have done this function enrichment and pathway analysis, and here you can see some of the pathways we have highlighted. So these highlighted pathways are associated with the hallmark of cancer. So after that, we wanted to see the association between these identified DEPs. So we have constructed a PPI network. So here you can see the 2 types of nodes. The red colors are representing the upregulated genes and green colors are representing the downregulated genes. So from this PPI network, we wanted to identify the key genes. So for that, we have done the module analysis and Harkin identification. And we got the 2 modules and the 66 Harkins. So here you can see some of the genes we have highlighted with the black color quantity. So these are the genes which are already reported as a lung cancer associated genes. So after identifying these key genes, we have done the pathway analysis and to see the impact of these DEPs on their this selected this pathway. We are showing here only the some selected pathway. So if we talk about this pathway in cancer, so here we can see that if we talk about this FGF gene, which is fibroblast growth factor gene, so we can see that this gene is actually involved in many signaling pathway like this MAPK signaling pathway, calcium signaling pathway, or the pathway which are later which are involved in the cell proliferation. So we can see that in lung cancer, this gene is down-regulated. So there are other genes which are down-regulated and some of the genes which are up-regulated in the lung cancer. So after this, we wanted to also see the association of these identified genes with the progress of the disease, and for that purpose, we have done the survival analysis. So we got the 4 genes which are showing the significant survival results. So here you can see that we can clearly see that the patient with the low expression of these genes are showing significantly better survival probability with the patient having a higher expression of these genes. So then we have validated the expression of these 4 genes in the TCGA database, and here you can see that all the 4 genes are upregulated in the lung cancer, which are also matching with our study. So we know, so these are the 4 genes, and now we know that this mRNA or the genes are also regulated by other biomolecules like microRNA or transcription factor. So we wanted to see if any of the microRNA or transcription factor is regulating the activities of these genes. So for that purpose, we have constructed this mRNA microRNA transcription factor network, and here we can clearly see that many of the microRNA are actually regulating the activities of these genes. And if we see, then this microRNA is actually regulating the activities of these all 4 DNA. And we also found out that this microRNA have been reported as a lung cancer associated microRNA, and it is observed to be and it was observed to be upregulated in the lung cancer. So coming to the conclusion, the key genes we have identified in our study could be used as a potential prognostic biomarker, and in future we would like to integrate other multi-omics data to better understand the mechanism of lung cancer and to make better prediction for the therapeutic targets. So these are the references. These are the references that we have used in our study. Thank you.
All right. So, Mohita, are you there? Can you hear us?
Yes, sir.
Oh, I first must say you have done too many things here in just one poster. So congratulations on that. Are there any questions for Mohita? Okay, if no, I have a question for you. This is kind of maybe a standard question is, you've done so much here and you're probably not the first one who's looking into integrating for lung cancer. So how does your result differ from the existing work that others have done? What is novel in your finding compared to the others, the earlier work?
Sir, if I have to talk about only the lung cancer, then it's very difficult to tell that I have done something new. But we have tried to identify the novel genes or pathway. But what we found that whatever genes we have found here in our study or the pathway, that somehow reported somewhere, maybe not the experimental, but bioinformatics, one of the gene we have identified, the recently one paper published, they were also talking about that this gene is upregulated in the lung cancer. So that's why we wanted to integrate others multi-omics data like methylation or the mutation rate, so that we could find the novelty in our work. So as of such, I cannot say the work I have done it's completely novel, but we are trying to find the novelty in our work.
Right. I think the multi-omics approach is perhaps the highlight of your work.
Yes.
And I think you probably have more data set than the earlier works because they were done in the past and you are in the present. So if you take as much data as possible and you do the same thing, and if you compare with what others have done, I'm sure you'll be able to find few nuggets of novelty, and then maybe you might want to consider publishing that if there's sufficient value there. Okay.
Yes, sir. Okay, thank you, sir.
There's a question here. Can we say novel biomarker as it is taken from dataset?
If we talk about for the lung cancer, then we can say that there's a potential biomarker, because we have seen like already the database curator database, we check that these identified genes are not considered as a biomarker for the lung cancer. So we can say that these are the novel biomarker. But we need the experimental confirmation also to validate our result.
There's a follow-up question. Have you done RT-PCR or any gene expression analysis?
No, we have done only the computational work. So RT-PCR we haven't done yet.
Okay, all right. Thank you very much. We move on to the next speaker. It's an oral presentation entitled SCID-Tuna, a network alignment approach for integrating multiple single-cell RNA-seq datasets by Yasin Maroof, Hilal Kazan, and Chezim Erten. Nihan, please go ahead.
Hello everyone, my name is Yasin Maroof. I'm a master's student at the Electrical and Computer Engineering Department, Institute of Science at Antalya Bilim University. I'd first like to thank the event organizers for the opportunity to present this work. So today I'm presenting SCID-Tuna, network alignment approach for integrating single-cell RNA-seq datasets. Single-cell RNA-seq technologies enable us to study the transcriptome at the cell level. The high resolution of single-cell RNA-seq helps highly heterogeneous cell populations identify new cell types, extract cell type-specific expression profiles, trace the lineage of cells, etc. Since its introduction 12 years ago in 2009, single-cell RNA-seq technologies have been constantly developing and improving the sequencing throughput, scalability, and speed. Efficient and up-to-date computational tools to analyze large-scale single-cell RNA-seq data are therefore needed to meet this growth of these technologies. There is a common need to integrate multiple single-cell RNA-seq data in cases where larger sizes are needed, data of from different tissues or under different conditions need to be analyzed together, etc. The problem with just concatenating data comes mainly from the batch effect, which is an effect that could hinder the detection of true biological signals of interest and could lead to inaccurate downstream analysis and misleading conclusions. There already exist multiple single-cell RNA-seq data integration methods that are developed to address this problem, such as Surat, single-cell align, and Batman. The integration process is a way of projecting two single-cell RNA-seq datasets or batches into a common space where the technical differences between them are mitigated and the true biological signals are more pronounced. The final result of the integration is expected to be a dataset where cells from both batches that are biological cells that are biologically similar clustered together while preserving the local neighborhood structure of cells. This is an example of a simulated data with two batches that have a small batch effect, and each batch has 3 cell types. We can clearly see on the left cells clustering not based on cell type identity only, but also based on batch identity. What we should expect is results similar to the ones on the right hand side. Let's now briefly introduce the SCID-Tuna algorithm. As inputs, read counts are first log normalized with a pseudo count of 1. Then the 2000 first or top variable genes of each batch are selected, resulting in 2 matrices, an N by G batch 1 matrix and an M by G batch 2 matrix, where N and M are the numbers of the cells in the 2 batches, and G is the number of genes. 3 graphs are constructed, 2 of which are reference graph G1 in the illustration and target graph G2, which we name intragraphs, and a complete bipartite graph, which we refer to as the intergraph, with one part containing the cells of the reference dataset as nodes, and the other part containing the cells of the target dataset. The weights of all the edges are the Euclidean distance in PCA space between the cells, and the intragraph edges, the black edges in the figure, are selected based on the Pearson correlation coefficient between the cells, where only the top correlated cells are connected. We use a thresholded minimum weights matching method to select reference target pairs, which are called anchors, which are the red edges in the toy example graph. For each reference cell X, an integration vector IV of X is initially set to be equal to the difference vector between X and Y being the matched cell of X in the log normalized gene expression space. Each integration vector IV of X is then updated based on a combination of X anchor weight, the red edge weight, the distance between X and its closest K neighbors, xn1 to xn4 in this example, the neighbors anchor weights, the blue edges weights, the distance between Y and the matching cells of the neighbors of X, yn1 to yn4 in this example, using the formula on the left. The updated IVs are then fed back to the formula to get new updated integration vectors, and this iterative feedback loop is run until convergence. To conclude the integration, the IVs are used to displace the reference cells by adding the integration vector of each cell to the cell vector in the original space. To evaluate the integration accuracy of our algorithm, we used 7 datasets, each having 2 batches, and used 4 metrics: local inverse Simpson's index, retained nearest neighbors, adjusted trend index, and the average silhouette coefficient. We are including only 3 of these datasets in this presentation. This is one of the simulated datasets, which is a balanced large batch effect dataset with 4 cell types. As a real dataset, we utilized 2 adult human hearts from a 52 years old male and a 47 years old female. This semi-real dataset here is a subset of a pancreatic dataset where only beta, delta, gamma, and endothelial cells are kept in both batches. Then gamma cells are omitted from the second one. From these UMAP plots, we can observe how well each tool integrated the ALB4CT batches. Well, we can see from these bar plots that Cytuna was the best performing method overall. We can see here that Cytuna was the only tool that could separate the gamma cells from the other clusters, the other cell types. Cytuna had the best Lisi score, adjusted rand index, and average silhouette coefficient, which led it to have the highest average score on this dataset. Cytuna integration of hard data batches looks much better than the other methods from these UMAP plots. Cytuna had superior Lisi and RNN scores and performed the best overall. We can observe that SCL-line has a low RNN score, not just in this data, but in all the other cases, which means that it has difficulty keeping the local neighborhood of cells intact. To assess that the integration of the batches did not disrupt the biological signal in the data, we compared the differentially expressed genes between each cell type and the rest of the cells before and after integration. The Jacquard index between the before integration set of DEX and the DEX in the integrated data is calculated and the higher this score or this index, the more decks are retained after integration. Cytuna had the best average score over all the pancreatic cell types, 73.8, followed by Batman with a score of 73.5, SCL line 56.7, then Surat with a very low score of 10.5. On the hard data, Cytuna and Batman had the same best average score of 48.2, preceding SCL line, which had the very close average score of 47.9, and Surat with the worst score again of 405.4. To summarize, we introduced Cytuna, a graph and anchor-based single-cell RNA-seq data integration algorithm. The integration accuracy of our tool was assessed by integrating the batches of 7 datasets and evaluating the integration results based on a local inverse Simpson's index, retained nearest neighbors, adjusted rand index, and average silhouette coefficients, and comparing the performance against Surat, Batman, and SCL line. Cytuna performance was in most cases better than the other methods. Finally, I would like to thank Professor Jasim Erten and Professor Hilal Kazan for their assistance and guidance. I would like to thank my Antalya computational biology lab mates as well for their help and encouragement, and thank Antalya Bilim University and TÜBİTAK for their sponsorship. And I would like to thank you for
All right. Thank you so much, Yasin. Are you there? Can you hear us?
Yes, I can hear you. You're welcome.
All right. Wonderful presentation. A lot of work. All right. The floor is open for questions. All right. So there's one question from Ms. Manjula Thima. How do your method differ from other graph-based methods?
So the one thing about our method is the last part where we do the iterative correction. So we use the neighborhood information of each cell to correct for the information of the matching. And how it is different or from MNN is we don't use MNN in our method. We just use another way of matching. So we don't look at the neighborhood at the beginning, but we use the neighborhood information as a correction information at the end of our algorithm.
Okay, I have a question.
Yeah, go ahead.
Awesome. Thank you for your nice presentation. That is a lot of work. Did you apply your data in the expression data sets, for example, for a disease data set?
Here we used so the pancreatic data wasn't a disease data set, but the second heart data was like a heart failure data set, as I recall.
No, on the expression data set. For the expression analysis.
For the expression analysis, it wasn't like a disease comparison. So it was like we tried to find the retention. So how many differentially expressed genes are retained before after we integrate. So if we observe a big change or the sets don't match before and after, it means that the integration disrupted this biological information.
Okay.
Yasin, I have a question. Am I correct to say that this is mostly focusing on batch effect in correction and integration, or it's beyond that?
So the initial step we are doing here is just between single cell RNA-seq data. So it is mostly the batch effect in between. So we're just trying to get rid of this technical problems and noises. But in the future, we are planning maybe to extend it into more multi-omics data that would be more beneficial, in my opinion.
Right, and you showed that there were 4 metrics that we used, and then eventually you use Jacket index. So would you say that, because having 4 different metrics can be quite challenging to evaluate overall, overall performance. So are you suggesting that with Jacket index, it's reasonable to use Jacket index as an overall evaluator, considering the 4 metrics that you had mentioned earlier?
So for integration in general, I mean, even in other omics data, the main metrics to use is this local inverse instance index and also a retained neighborhood. And for this specifically for single cell RNA-seq data, I mean, it made sense to also look into differentially expressed genes. That's why we added that one. But I don't know. I mean, I cannot confirm that it is the one that wrap up all the evaluation here.
So is there such a metric that could be used for overall performance? Or how do you handle a problem where you have to look at overall performance?
So for now, I don't think it exists. So for all the benchmark work in the literature, I see that they use multiple metrics. So it's a combination of both, a lot of metrics. It's almost like we can say that it depends on the need of the integration. So it depends on what's the objective of the integration. If we should focus on separating cells or having a good integration of the batches. So it depends, but we don't have I don't think there is a single metric used to evaluate the whole integration.
All right. In the interest of time, I'm gonna stop here for yours. And there's still one question from Manjula, which you can answer through the chat. Thank you very much, Yasin. Very good work. All the best to you. Moving on to the last presentation. Sure. Last presentation is a poster presentation. Evo Lirova, an artificial intelligence program for maintaining health condition. This is by Viona Priscilla and Nanda Rizkia Paradana Ratnasari. Nihan, please go ahead.
Good day, dear judges and everyone. I am Fiona Priscilla alongside with Miss Nanda Rizkia. Today, I would like to present the innovation on preventing diabetes mellitus, especially type 2 diabetes mellitus. Let's understand more about type 2 diabetes mellitus, or also known as T2DM. Type 2 diabetes mellitus is a chronic metabolic disease characterized by elevated levels of blood glucose. The disease can occur through insulin resistance and/or impaired insulin secretion. Insulin resistance causes normal levels of insulin to become less effective, which calls for the need of more secretion in larger quantities. The continuous exertion will cause beta cells dysfunction and subsequently impair the insulin secretion. The most common clinical outcome of type 2 diabetes is obesity or a higher body fat percentage, typically distributed in the abdominal region. Moreover, the disease can lead to complications, including cardiovascular, cerebrovascular, and peripheral vascular diseases. In 2020, the prevalence of diabetic patients in Indonesia reached almost 11 million people, and by 2040, it is estimated that the prevalence of diabetic patients in Indonesia will increase and reach 16.2 million people. Therefore, Lirova has a target to decrease this number. In order to educate people to do a healthier lifestyle, starting from the diet for manage the calories intake, Lirova is a dietary calculator that is packed with a personal assistant or AI. Therefore, please meet IFO the AI. In Lirova, IFO will provide the users all of the Lirova application services, which consist of BMI, BMR, and set reminder room. But before that, IFO needs to record the user's personal data such as username, height, weight, gender, and age, especially for the new users. This data is only asked once, but if the users make a mistake, they still can revise it. LIROVA applications were designed as simple as possible to give users the best services where the users only need to interact with IFO, our AI in the chat box. Of course, this step can only be done after the users register their personal data. In order to make it effective, the main menu contains a few features only, but each function was very beneficial. As mentioned before, the user still can revise their data in the profile page on the main menu. This action will affect the interpretation of the results, which will be saved on the history page. Therefore, LIROVA will recommend the user to update their data once they are really having some changes. In the About page, it will cover any frequently asked questions to make it easier for users to reach the LIROVA development team if there is any uncertainty. Further research is still to be done in order to validate the interpretation results from LIROVA. Together with LIROVA, we prevent the spread of T2DM. Thank you for the attention. LIROVA prevents diabetes only from a chat box.
Thank you so much. Fiona, are you there?
Yes, Doctor.
All right, great. Questions for Fiona from the floor? Anyone? All right, if no questions, I have one question. Am I correct to say, Fiona, that your Evo LIROVA is mostly focusing on educating the masses through this app, and you're using AI to kind of understand the habits of, the population and then make recommendations? Is that correct?
Yes.
So what do you think? Why do you think people would really care, to spend their time on this app? What is the attractiveness? I mean, what's the incentive? I know people are aware about health, but even then people don't do much. How would you attract people to come on board?
Okay, thank you, Dr. Khan, for the question. I will try to answer it. So basically, because that one is our first awareness to create this application. That is why I have mentioned on the previous that I create this application as simple as possible to make it the users not get any confusion in the preventing diabetes by using only the chat box, and for users to attract the users to use our application is only just some education and maybe just sharing something related in the related event. I just planning to be publish it, something like that. Yeah.
All right, my advice would be to tie up with the Ministry of Health, because a top-down approach and bottom-up approach that might give you some traction. On the other hand, I think if people see no value besides advice and education, I doubt they would stay long. So it may not be sustainable in the long run. You might get people come in, but they may not stay long, or they may not use it as much. So, I think you need to think about how to make it valuable. If they see value, then you would attract. So value could be in terms of, personalizing the diet, recommending diet in terms of what they're, taking in and what they could cut, something along those lines, because I see there's not much understanding or awareness of what they could consume, what they couldn't consume, especially for the masses. I think that could be one area where you can actually contribute. If your system could see what is their existing diet and how you could make a recommendation of improvement, but you got to be careful because, you want to make sure that this is certified by a nutritionist. Anyway, these are some of the things to think about. It's a great effort. Well done. And I hope many people sign up and listen and do something so we can cut down, the spike of the T2DM that's expected. 6 million going up, that's really a lot. So I hope, I wish you all the best. All right.
we can use the feedback to improve our paper as well. Thank you very much.
Sure, thank you. All right, with that, we are done with this session on biotools, big data. Thank you everyone for your attention. Apologies for the hiccup earlier. It seems like the other room kind of finished earlier. So because of that, when they were exiting, it kind of cut off all of us. But in any way, this is good because now more people could listen to the talk. So that's, I guess, blessing in disguise. Thank you for your patience. I know we have much shorter break. So I pass it back to you, Nihat.
Okay. Thank you, Professor Khan and our honorable judges. I would like to thank all our participants for being here with us. We will now have the lunch break. We will meet at 2 p.m. Turkey Standard Time. The participants are invited to attend our next parallel session. Then we will have a keynote talk later in the day by Dr. Yuval Itan at 4 p.m. Turkey Standard Time. Thank you and enjoy your break.
Welcome back, everyone. We will again be going for the parallel sessions. Please join your desired rooms. All right, everyone, welcome back. So I hope you have had a fruitful scientific session. And now we are waiting for Dr. Yuval Itan as our next keynote speaker to join. So please stay tuned. Thank you. Hello again, dear all. I am sorry to announce that Dr. Yuval Itan is not going to be able to attend today's session. So see you tomorrow. I hope you had a fruitful scientific session. Due to the emergencies that we couldn't handle, we had to stop here and then continue tomorrow. Hope to see you tomorrow again soon. Good evening. And also, I would like to tell you before we leave about our feedback reports. Please check the link through your chat box. We would like to get feedbacks from you. Thank you.